All right, we're going to get started promptly today. You may have noticed on the fire drill schedule that Building 3 is supposed to happen at 11:45. We've asked them to delay, but be warned that at any moment there might be very loud noises and we will have to evacuate. So we'll go straight to David Damby's talk. So we welcome David today. He's harkens from Northern Illinois. He did a bachelor's degree at the University of Vermont in biochemistry and actually did some research in the Department of Transplant Surgery. So he comes from a great breadth of background. But then he decided that he liked mountains more than the lab, as many of us geologists have decided. So then he went to Durham to do a PhD that was in partnership with University of Edinburgh lung research, but in earth science, working on the health hazards of volcanic ash. Before coming here, he did a postdoc at Munich at LMU in experimental volcanology with Don Dingwall, and he arrived here at the USGS in Menlo Park back this past summer, and he's working in NRP, but on volcanic hazards, on the health and environmental hazards of volcanic emissions. So please welcome David.
Thanks a lot, Hannah, and thank you all for coming. It was my hope when putting this talk together that I could come up with a title with sufficient intrigue that I might lure you out of your office for 45 minutes or an hour. So I'm glad to see you all. Thanks for coming. And also, while putting together this talk, I was trying to figure out a way to show you what the interface of medical geology looks like. That is, how far into both fields we go, and how there's really a continuum between them. But I also wanted to showcase the breadth of things that medical geology entails. And so I think I found a balance by offering you one story about crystalline silica. It's a mineral we all know and love, and then I'm gonna give you a couple other stories, but just the Cliff Notes version, so just really brief, so you can see some of the other things that we do. And then at the end, I'm going to put that all together to compile a picture of what the volcanic ash hazard looks like, and then raise that again to medical geology to show you why I think anyway this kind of work should be done within the USGS. So the first story then, the crystalline silica Enigma, and I apologize for these titles. The second story, the secret life of ash. Then we move on to Iron Iroth, and finally, what's bugging you? A title thanks to Jess. So to start then, the crystalline silica Enigma. Now this story starts, as so many do, with the 1980 eruption of Mount St. Helens, which led to widespread ash deposition in the Pacific Northwest and farther inland, and resulted in a bunch of people walking around looking like this. And that, for us, signifies a public health crisis. So following this eruption, people started to ask, is volcanic ash dangerous to your health? And the reason they started asking this question, well, one, because it's a particulate that you can inhale, but two, because it contains crystalline silica. And crystalline silica is a toxic mineral. And it turns out that it's the only toxic mineral in volcanic ash. But during this eruption, there was a lot of controversy in quantifying crystalline silica. And you can see at the bottom there, depending on which lab you speak to, it could range anywhere from about 1 to 12 weight percent. And 1 weight percent is not such a big deal. We don't need to worry about it really. But 12 weight percent is in the realm of what we see in coal dust. And as many of you may know, coal dust is an absolute nightmare from a public health point of view. So when we talk about the toxicity of crystalline silica, what I mean is that it has a well-established disease endpoint. Exposure to crystalline silica results in the disease silicosis. It's also a carcinogen. It's got a group 1 rating, which is the highest that the IARC, the International Association for Research on Cancer, can classify things as, and it shares the company of things like asbestos, plutonium, right? These things that we know are pretty bad for you. It's so reliably toxic that it's used as a positive control in all sorts of toxicity experiments. And I just showed you at Mount St. Helens up to maybe 12 weight percent, but in work we've done since, we've seen up to about 25 weight percent in volcanic ash. So that's a lot. About a quarter of what people can be exposed to is this known toxic mineral phase. But there wouldn't be an Enigma if there wasn't a caveat to that. And in 1997, the IARC, when classifying crystalline silica as a group 1 carcinogen, said that carcinogenicity to humans was not detected in all circumstances. And it's classically an occupational disease. And so all of the literature is from the industry, which is why they say industrial circumstances. And they go on to say that the carcinogenicity may be dependent on inherent characteristics as well as external factors. A year later, talking about silica-associated disease in general, my old supervisor Ken Donaldson and a colleague Paul Borm went on to say that the hazard posed by quartz is not a constant entity, but one that can vary dramatically depending on the origin of the silica, as well as its contact or presence with other things. And if that sounds a bit vague to you, I agree, because effectively all they've both said is that anything that can be different is probably responsible for causing a difference. So what we want to know is, does this apply to volcanic crystalline silica, right? This ash that people are exposed to. Do we need to worry about whether or not people can get disease from exposure? So this is unraveling the crystalline silica enigma, as it is for volcanic ash. And just by way of a brief outline, we'll talk about the origin of crystalline silica in ash, right? How it gets there, what's the source of this hazard. Then we'll have a look at the biology side. We're going to go in depth, but don't worry, I'm here with you. And we'll see what the actual response is. Then we'll take a look at the mineralogy of crystalline silica and how that governs the actual response that we see. And then through some experimental work, we'll see if we can provide some insight into whether or not volcanic crystalline silica is actually toxic. So we can get crystalline silica in ash through a couple of different ways, really. I mean, it can be as a primary phenocrystalline as quartz that ascends with the magma. But it can also form in volcanic domes and volcanic lavas through a couple of different mechanisms. And when it does this, it forms as cristobalite, which is a secondary mineral. And it can happen by two ways. One is vapor phase crystallization, which is this image you see in the top right. These are SEM images of cristobalite crystals in pore space in volcanic rock. And you can see nice big euhedral crystals hanging onto the side of the wall. And you can also get it through devitrification of the ground mass class. And that's the picture in the bottom right there, where this is an SEM backscatter image. You can see all the dark patches just in the center there. That's all cristobalite. The black space is void space, and you see some microlights and some ground mass glass in that as well. And that makes volcanic domes really a unique hazard because they can mass generate crystalline silica as cristobalite, right? It's this addition of crystalline silica that we don't see in primary magma. Just to show you a couple of, I think, beautiful images of what it looks like in situ. This is for vapor phase crystals. On the top, you have these nice, well-formed euhedral crystals. And then on the bottom, you have the second morphology, which are plates. And I'll get on to why this may or may not be, but this is what the vapor phase crystals look like. And then when you look at devitrification, it also has a couple of different habits. The top left, again, sorry, these are all backscatter SEM images. The black is all pore space throughout. All of the dark blotches in, well, all of the images are cristobalite. And so in the upper left, you can see just these little spots that are pretty pervasive throughout the ground mass. In the upper right, you can see these really concentrated, very pure-looking patches. You can see some overprinting in the bottom left. This is the image I showed you before. And then you can also get a range of textures in the bottom right there. Again, the dark is all cristobalite. The light gray, for example, this patch over here, that's all ground mass glass. And you can see, I think, pretty convincingly that the cristobalite is devitrifying into this space. Now, I said before that quantifying and characterizing cristobalite in ash was quite difficult in the wake of Mount St. Helens. And it still proves to be a little bit, but we've got a much better handle on it now. And so the way that I went about doing it was a combined use of Raman, backscatter, SEM imaging, CL detection, and chemical mapping with EDS. And I hope quite convincingly you can see that, in fact, everything we're dealing with is cristobalite. And it turns out that the abundance of cristobalite in dome rock is a function of, I say, residence time and volcanic activity. And so you have the evolution of a hazard. When magma ascends, that sort of time is 0, and then with time you have an increasing abundance of cristobalite. What that looks like, if you look at the top two images, the upper left is just the very start of crystallization. You can see some nucleation happening, and in time you get to this image in the upper right. Again, all of these dark blotches are the cristobalite. So you can see almost, not complete, but relatively complete replacement of the glass with ground mass cristobalite. And if you look at the bottom two figures, on the bottom left, you have some data from Mount St. Helens. And you see that with increasing dome residence time, this is all calculated by the USGS, you see increasing cristobalite concentrations, which, again, is a reflection of this idea of residence time. But if you look at the right, this is data from Soufriere Hills, you can see two lines that I've drawn. The top, more flat, downward sloping line is for domes that are slowly extruding, whereas the bottom upward sloping line is for domes that are more quickly extruding. And so when I say residence time, I'm not talking about start the clock, stop the clock. I'm talking about time spent within some PT condition that allows crystallization to happen. And when you think of vapor phase cristobalite, you need a source of Si. And that source can come from two different places. It can either be local or from depth, right, from outside of the immediate vicinity. And so again, if we plot up this data from Mount St. Helens, we can put together a bit of a theoretical model on where this Si comes from. And so if you look at the plot on the right, you have bulk SiO2 by XRF, and then on the y-axis, you have the abundance of cristobalite. And in the lower left, as you would expect, there's not a lot of cristobalite, low Si chemistry. As you get to samples that are more dominated by devitrification cristobalite, you see this increase in cristobalite, but not much of a change in bulk chemistry. Whereas if you look at the third envelope on the upper right, you see in samples that are dominated by vapor phase cristobalite, you see this increase in Si. So that to us sort of suggests that whilst some Si may be sourced locally, you can also have transport through the system and deposition once you reach saturation in a certain PT window within the dome. So what we know from all this essentially is that this is through studying a bunch of different dome rock at a bunch of different volcanoes listed across the bottom there, a bunch of well-known ones, Mount St. Helens, Colima, Sant'Agito, right? That cristobalite isn't specific to one volcanic center. It can form in everywhere we've ever looked. And each location can have a range. But what we've been able to do is, through identifying the source, the origin of cristobalite in a dome, is understand how this hazard changes. And that's really important when you want to talk about the hazard posed by ash, right? So having seen a bit of the origin, what's it look like when we actually put ash into a lung? Now, when you breathe in ash, ash containing crystalline silica, well, all ash in general, in fact, anything you breathe in, the first thing it encounters is the lungs. And this is what the lungs look like. If you look at the two images on the right, this is a lung that's been impregnated with resin, as we do with rocks, and sectioned, as we do with rocks, and then imaged by microscope, as we're all familiar with. And all of the white, the grayish, is open pore space, right? This is the part of the lungs that we actually breathe into. And all of the pink is tissue. And if we zoom up on some of the tissue, in the bottom right image there, we can see that there's a layer on top called the epithelium, and that's the skin of the lungs. And on that, you have this cell called a macrophage, and they're responsible for defense. They patrol this epithelium and take care of any particle that might come in. And so if we're talking about whether or not ash is a hazard, they're a great place to start. Right? What's the response of a macrophage to ash? And so this is exactly what we do. In the upper left, all of these circles that look like bubbles, those are cells, those are macrophages. And then in the bottom left, all of the dark circles are macrophages that have been treated with ash. And so what happens is these cells go around, gobble up all the ash, internalize it, and that's what we're seeing. But if we look at the biochemical response, not a whole lot happens. On the right here, viable is the amount of cells that are still alive, and you can see that it's all about 100%. Necrotic is the amount of cells that are dead, and you can see it's all down around 0. So not much is happening. So we looked a little bit further, and apoptotic is a measure of whether or not they're a bit suicidal, I guess. Not necessarily dead, but not in such a good state. And again, you can see pretty much 0. Not a whole lot's happening. So we said, okay, well, they're alive, therefore not dead, not even thinking about it. Are they under any stress at all? And that's what this last column is. It's an oxidative stress marker. And again, we see nothing. So they're really quite happy, these macrophages, after they internalize ash. But I told you at the start that crystalline silica is so reactive it's used as a positive control in toxicity experiments like this one. And in this ash, we have upwards of 25 weight % crystalline silica. So what's going on? Well, crystalline silica enigma, right? We looked a little bit farther, and we said, okay, not dead, still alive, not stressed out. Are they doing anything at all? And this marker on the y-axis, IL-1 beta, is a measure of, are they talking to each other, right? They're happy, but are they at least communicating and saying, hey, there's something here that's not supposed to be here? And indeed they are. That's what we see. And again, a range of samples from a range of volcanoes along the bottom axis there. And DQ12, second from right, is a positive quartz control. So you can see it's behaving as it should. And what we can see is that the production of IL-1 beta is sample dependent, right? So not all ash is the same. We wanted to know what's responsible for this production of IL-1 beta, right? Is it our crystalline silica, or is it something else, right? Because that's the key question here. And so we did a bit of an old school componentry analysis where we synthesized or sourced the pure phase componentry of ash, that's present in any reasonable quantity at least. And we did that through remelting of an andesite, sourcing labradorite from the state collection, converting quartz into cristobalite at high temperature, and then we also used the crushed pumice sample as a cristobalite-free material control. So that there are some things here that we didn't account for, but hopefully this accounts for all of the things like oxides or whatever. And what we see, sorry, just on the lower left is a grain size distribution of that, showing all of the samples and a nice small particle size, which is what you need when you work in the lungs. And what we see, if you look on the right, this is IL-1 beta again, right? This is this communication marker. We can see that cristobalite really is the only one that's causing much of a response. And so from this, we say that cristobalite indeed is the mineral phase that's responsible for what we see. Now, a lot happens between when we sprinkle ash on cells and we measure IL-1 beta, right, this communication marker. And it looks like this. We have a crystalline material, and then this purple curve is the cell membrane, right? And then what's below it is all of the signaling that happens inside of a cell. And IL-1 beta is produced, not always, but often, through a complex called the inflammasome. And it's a bunch of proteins that get together and cleave pro-IL-1 beta on the lower right, this precursor molecule, into active IL-1 beta. And then that's what actually gets released. And so what we did was we wanted to know if this is actually the mechanism by which we see cells communicating. It's one that's been identified for a couple of other things, both environmental and a lot of different exposures. Things like uric acid and gout, cholesterol and atherosclerosis, and then also exogenous things like asbestos and crystalline silica. So we figured if this pathway is responsible, we've already identified cristobalite in the last slide as the responsible agent. This is sort of the confirmation that indeed it is working by the way we expect it to. And so we put volcanic ash into this model. And the way we did this was by taking this protein complex and selectively deleting one at a time, so that in these cells that have been genetically modified and are missing these components, without them they can't produce IL-1 beta. And effectively, well, not effectively, what we do see is that if we delete any one of these proteins, we no longer see a response. So the black is our normal system. And then the white, the gray, and the striped are all when we delete a protein. And you can see that deleting any one of them completely gets rid of the response. And so we're confident to say that the IL-1 beta, right, this talk that we see between cells in response to ASH, is happening by this mechanism. So what does this mean then for the hazard, right, for what happens in the lung? Because I showed you that we don't see much of a response, but I've also told you that crystalline silica is reactive. And so again, these are cross-sections of lungs. This time it's black-gray rather than pink and purple, but the white is still the void space and the black is the tissue. And the left two images, top and bottom, are a normal system, right? The top is the lung, the bottom is the lymph node. And that's what happens, the lymph is the spot in the body where ash or crystalline silica goes after you breathe it in. And so that's a healthy looking lung. In the middle is a cristobalite standard, right? A positive control, this thing we know that is reliably toxic. And you can see thickening, a lot of reduction in pore space. This is all bad. This is what causes you from breathing normally to having very labored breathing. There's just not enough space. And in the upper right, when we treat with volcanic ash, it doesn't look a whole lot different than the normal system. So even though we're seeing this talk, not a lot's happening. But when we look at the lymph, you can see the response to cristobalite. You get this massive clustering. And we see the same massive clustering in volcanic ash. And so what we're saying is that even though macrophages are quite happy to handle this and get rid of this, and they don't die, nothing happens, the conversation that's initiated by ash through this signaling mechanism results in delayed problems, right? This problem later in the lymph. So what is it about volcanic cristobalite that's causing this non-conventional response to crystalline silica? This is where we go back to geology. We came up with a couple of theories about why this decreased reactivity could be happening. The first is that particles, ash, right, is a mixture of phases. We know that. There's cristobalite, but only a quarter of it or less. So there's other things in it. Second theory is that there's some sort of coating around the crystalline silica, right? There's a very unique history to volcanic eruptions, gases, temperatures, so on and so forth, that could impart some sort of amorphous coating on the grains. We thought the silica particles might be chemically impure, or the silica particles might be structurally imperfect. And so the way that these all work is, again, ash is a mix of phases. And so if we take cross-sections of volcanic ash and look at it, backscatter SEM images, we can see that indeed some particles, like in A, are pure cristobalite, whereas other particles aren't, right? In B, you have a crystal hanging onto the edge. In C, you have this core of cristobalite, but a very heterogeneous rim of ground mass around it. And same with D, where it's mostly cristobalite, yet you have this fine-grained rim around it. And so what we know is that 100% of the surface of ash is not crystalline silica. So that might be responsible for the reduced response that we see. To this idea then of an occluded surface, we took a high-resolution TEM transect of the chemistry and structure across a cristobalite particle, and we didn't see any difference. There was no change in chemistry or structure from the core of the particle to the rim of the particle. So we concluded that this is not likely to be the case. So the idea of crystal chemistry then, for this we went to the microprobe and did a whole bunch of spot analyses from a whole bunch of volcanoes. And what we see is that, yes, indeed, cristobalite contains aluminum, up to about 3 weight %. And all cristobalite we've ever analyzed shows aluminum impurities. And for reference in the bottom right there, these are all quartz samples. So the level of impurities that we see are quite different. And lastly, what about structural imperfection in this crystal? Well, because ash is a mixed phase, getting structural refinement for one of the phases is quite difficult because there's a lot of interferences. And so what we did was use the transition of cristobalite from a low temperature form alpha to a high temperature form beta as a proxy for structure. Because we know that at lower temperatures, this mineral undergoes a transition, with a more imperfect crystal undergoes this transition at a lower temperature. And it's quite a big range, from 170 to 200 degrees centigrade. And what we see is that volcanic cristobalite, this blue curve on the left, does indeed undergo this transition at a lower temperature than a more pure phase synthetic sample. And so we're also on board with this theory of it having an imperfect structure. And to summarize those, just again, we have impure particles, no evidence of an occluded surface, we have these cation substitutions, and we have a poorly ordered structure. And these cation substitutions are important because aluminum is known to inhibit crystalline silica toxicity. But at this point, they're all observations and they're all theories as to whether or not they can actually impart an effect on the response. And so we did some experimental work to see whether or not these theories could indeed alter the reactivity. So to get at this question of structure and chemistry, we had to take a two-prong approach because cristobalite, it's a metastable mineral. It forms at high temperatures. You can see in the upper left between 1470 or so and higher, which is not volcanic environments. And so in order to look at the effect of structure, we had to synthesize cristobalite at high temperatures, at 1400, 1500, 1600 degrees. And the result of this is lower formation temperatures have a less perfectly ordered structure. And so we use these high temperature samples as to inform the question of structure. And then we were looking at low temperature synthesis and wanted to introduce impurities. So we synthesized cristobalite at dome relevant temperatures with 1, 2, and 3 weight percent of aluminum. And this was to directly answer the question of do the substitutions play a role. This was all done at the same temperature, so there's no structural effects imparted by formation temperature. And what we see is that, yeah, they both do. The structurally less perfect cristobalite, this high temperature stuff, if you look at the image on the left, the sample done at 1400 versus the sample done at 1600 shows lower toxicity. In this case, we're talking about this communication marker again. So the more imperfect structure incites less conversation. And if we look at the chemistry, which is on the right here, we can see along the x-axis increasing weight percents of impurities, and then LDH release on the y-axis. This is a marker for death. And we can see that as we decrease, so as we move towards 1 and 0, we see a difference between the high aluminum content samples. And so we conclude from both of these experiments that both of these theories do actually impart an effect on the crystalline silica reactivity. So what about this question then of of a heterogeneous surface, right? That ash is part of a mixed phase, crystalline silica is hosted by a mixed phase particle. And to do this, we made our own ash. And so this is a picture of the setup here, where we core a sample, and then through decompression, we fragment it and generate and collect our ash. And what we see is that this also has an effect. In the plot, you have 5 different scenarios along the x-axis. This is decompressed from root temperature, from high temperature, milled, abraded to represent a pyroclastic density current, and just a natural ash sample. And the blocks all represent the percentage of surface occluded. And what I want you to take away from this is that it's different, right? So the mechanism by which we generate ash results in different occlusion of cristobalite at the surface. So those 3 theories that we talked about, heterogeneous particles, chemical impurities, and structural imperfection, do all actually play a role in the decreased reactivity that we're seeing. And so I want to remind you of this idea of inherent characteristics and external factors, right? This idea that anything that could be different probably plays a result. And so through some experimental work, we've been able to constrain what those factors are, right? We have a heterogeneous particle, and then we have these substitutions as well as structural imperfections. And I think what's important to note from the geology side of this, regarding these aluminum substitutions, is that it's present in all cristobalite we've seen. Right? We've never analyzed a cristobalite crystal that doesn't have aluminum. And also through our synthesis experiments, we only saw that we could form cristobalite by doping it with aluminum. And so my thought is that this same element, right, aluminum, that permits cristobalite to persist metastably in a volcanic environment is the same thing that results in its decreased reactivity. So at this point you may say, well, that's the crystalline silica enigma done and dusted, right? We've got it all figured out. So we took those same ideas and applied them to different exposure scenarios. And we'll give you a brief on three of them. The first is diatomaceous earth workers. And diatomaceous earth, when you process it, you introduce cristobalite. And the epidemiology literature says that there's a high incidence of disease amongst diatomaceous earth workers. And so we looked at the particles to see if we could figure it out. And effectively, there's no relationship between crystalline silica content and disease incidence. And so this is one that's still an open question. So this whole crystalline silica enigma thing, we've got it sorted out for one scenario, but it doesn't directly translate to another. Another case happening right now, it's of very recent interest, is silicate-associated osteoporosis, which is quite prevalent in California. And apologies for the perhaps graphic nature of the image, but you can see the bowing of this horse's back. And that's what people at Davis think. They think silica is responsible for this. And the reason they think that is because if you look at the X-ray in the bottom, this is a chest X-ray of a horse with the ribs kind of vertically, just barely visible. You can see a lot of cloudiness, and that's all respiratory disease. And these things come in tandem, right? This osteoporosis and this respiratory disease. But this is unique because it's not something we also see in humans. Humans with silica-associated disease don't have the same incidence of bone weakening. The third case, and I'll warn you now, there's a bit of a graphic image on the next slide, so look down if you want. You won't miss anything by not seeing it. Is a case of non-industrial pneumoconiosis, which is a general term for lung disease, well, a very specific term for lung disease, in northern India. And there's no industry at all in this region of India, but there's a very, very high incidence of lung disease. And what we think is it's all due to exposure to the regional geology. But on the lower left here, you can see a bunch of particles. This is ChemScan data, mapping phases. And we don't see a correlation between data from that and the toxicity data. Right on the right here, you have a diseased lung. On the bottom, in the bottom left corner, that's what a healthy lung looks like. In the top right, that's a tumor. And so in this area, at the moment, we aren't able to relate these two things. So even though we've got it figured out, we think pretty much for volcanic crystalline silica, it doesn't mean that we have it figured out everywhere. And so I'd suggest this sort of reinforces the idea of inherent characteristics and external factors, or whatever that may mean. So that's the crystalline silica story. Moving on then, again, I'll just give you these in brief. A lot can happen to ash between an eruption and exposure. Right? There's a big history from the eruption dynamics themselves, as I showed you previously, to transport in the plume, processing in the atmosphere, processing in the environment, and then also processing in the body after deposition. And so I want to talk just quickly about what each of these things look like. And we'll start off at the end of that with processing inside of the body. And to illustrate this, we have a sample from Colima and a sample from Merapi, and both these samples contain an abundance of cristobalite. And we took them and we digested them in synthetic lung fluid. Right? It's a chemical mix, like Gatorade almost, which mimics what the lining of your lung, the fluid in the lining of your lung looks like. And we did this for 4 weeks and then analyzed the mineralogy afterwards. And what we can see is in the case of Colima, this peak you see, this just raw data at 22 degrees, before and after is the blue curve, Colima gambles, you see no difference in the peak intensity, right? So that tells us that there's no change in the relative amount of cristobalite to other things. Whereas if you look at the right, this case of Merapi, the original sample in red and after digestion in black, you can see an increase in this peak height. And what that tells us is that you have an enrichment in crystalline silica. And so 2 different samples, generally thought of as ash, certainly by the biology community, have 2 different behaviors after exposure. And so this is saying that some ash, the expected behavior doesn't change from the long-term behavior. But for other ash, what's deposited in the lungs may not be the same after days or weeks or months. And so the exposure that you're experiencing in situ may change with time. Now, exposure doesn't always happen to just ash, right? A lot of volcanoes are situated very, very close to big cities. I guess flip that around, right? Big cities are situated very, very close to volcanoes. And so you often have co-exposures of the ash plus whatever's in the atmosphere at the time, right? Anthropogenic pollution being a big one. And so what a student of ours was looking at is co-exposures of ash with exhaust. And that's what you see here. TNF-alpha, this is another one of these communicating signals to see whether or not cells are talking. And you start with a negative control and then volcanic ash. REVA is a repeat exposure of volcanic ash. And then DEP is diesel exhaust. And you can see that when you have these 2 combined exposure, DEP and VA and DEP and REVA, you see an increase in response. And so what's happening to ash only, right, this whole first section that I told you, isn't always the case, right, because it can be augmented by exposure with other things. And just in the bottom right there, this is a system that we haven't used yet, but it's their setup where they have a car plugged into a chamber with cells. And so they essentially just turn the car on, start it running, and let exhaust pass over the cells. So we haven't used it yet, but we're looking to, and we'll see what happens. And then lastly, more towards the initial ejection of material, volcanic plumes are pretty complex. And so it's likely that plume processing of ash has an effect on these particles. And I've outlined a couple of things that could be happening here, right? Known factors that reduce crystalline silica reactivity, and as I said, presence of aluminum, but also adsorption and etching by acids, which is undoubtedly happening, high temperature treatment, as well as adsorption of water. So all of these things that are very common in volcanic plume can affect crystalline silica reactivity. And so the way that we're getting at this experimentally is by processing ash in a reactor which simulates a volcanic environment. You can see the cocktail of gases here on the left, CO2, HCl, SO2, H2O. There's no HF in the system yet because it will completely destroy all of the lines. So that's going to be like a final hurrah once we figure out the effects of the chemistry on all these other things. So that's just in brief, right? What I showed you at the start is the case for ash, yes, but maybe not always, because a lot of things can happen to the ash surface that may affect the reactivity that we see. Iron IROF, and I apologize for that. The other hazard posed by volcanic ash, in addition to crystalline silica, or the other mechanism by which volcanic ash can be reactive, in addition to crystalline silica, is through surface iron. And this happens in the body through some redox chemistry, whereby surface iron, in the presence of hydrogen peroxide, can result in a hydroxyl radical. This thing over here that's underlined. And hydroxyl radicals are highly reactive and very unstable. So any biomolecule they interact with, they destroy. And it's this sort of mechanism is implicated in all sorts of things like the origin of cancer. And what we see is, well, the good thing is that we can measure this in the lab, right? We can set up an experiment to measure whether or not ash is capable of producing radicals. And so along the x-axis, we have, again, just a bunch of different volcanoes, well, a bunch of samples from a bunch of different volcanoes, this smattering. And then on the y-axis, hydroxyl radical production. And we can see that there is no standard level, right? Samples from the same volcano can produce different amounts. Samples from different volcanoes can certainly produce different amounts. And so there's no way to predict this. And because this reaction is done in the lab, we can control it so it's only specific to iron. So in theory, hydroxyl radical production should be related to the content of iron on the surface. But we don't see any correlation at all. In the top image here, you have, again, radical production on the Y across bulk iron, right? So this is the whole sample. And we don't see any relationship, outside of maybe high iron contents. And on the bottom, again, radical generation against iron release. So this is surface iron that we measure through chelation, right? We just pluck it off, and then we can count up how much iron is only on the surface rather than in the bulk. And again, what we see outside of some of these basaltic samples, in this purple square rectangle, there's really no correlation. So it matches with the graph on the top, right? These high iron content samples do tend to produce more radicals, but beyond that, there's not a whole lot of correlation. And so unlike the crystalline silica enigma, which we think we've got kind of figured out, this question of iron is something that's still completely up in the air. But similarly, we took an experimental approach. And again, this is fragmentation of rock to generate our own ash samples under a multitude of different conditions. Some of the acronyms there are high temperature for HT, RT is room temperature, and then different pressure conditions that we decompress from, whether or not sample's crushed afterwards or if it's straight from the experiment. And what we can see, if you look at the iron box, is that the mechanism by which we generate ash has an effect on the surface iron. On the y-axis here, this is a ratio of surface to bulk iron, and the surface iron in this case is measured directly by XPS, so it's a chemical analysis of the surface. And we can see that we can change the ratio, right, the presentation of iron at the surface based on how ash is generated. And the other thing that we can do is put this ash in the plume that I showed you, this plume simulator, this reactor, and what we can see is that the history of the ash in this gas mix affects iron solubility at the surface. And so on the left box is a control, time on the x-axis versus iron released on the y-axis. At 300 degrees in a nice cocktail, we see one release kinetic, and at higher temperature with the same cocktail, we see a different release kinetic. And so we can say that much like we expect plume processing to affect crystalline silica reactivity, we expect plume processing to affect certainly the bioavailability of iron, if not also the particle surface reactivity. To wrap up then, my hope here was that in showing you this, these three final stories in addition to the crystalline silica enigma, in addition to having promised them to you, was that I could show you that I probably have some common ground with everybody in this room. And so this last one is about bacteria and volcanic soils. And so if that's your thing, I'd love to talk, because this work is in its infancy and needs a lot of help. What we've seen is two things when it comes to volcanic ash and bacteria. The first is that when you're exposed to volcanic ash, your body is less able to handle infection. And so that's this plot on the lower left. Bacterial killing is a macrophage's ability to kill bacteria. And when you're exposed to a control versus ash exposure, you can see decreased killing potential for macrophages. And so this has a lot of implications because bacterial infections are very widespread. And places like Indonesia, which also has quite a bit of ash, you have a very high incidence of pulmonary tuberculosis. Now, I'm not saying that they're directly correlated by any means, but I'm saying that in and amongst other things like smoking, socioeconomic factors, it's something to consider. And the other thing that we see is that volcanic ash, when used as a substrate, is very fertile in terms of bacterial proliferation. And that's what you see in the lower right here, where bacterial proliferation in this case is signified by OTUs. Volcanic ash, or volcanic substrates in general, the ash and the glass, have a lot more total OTUs than the other more conventional substrates. And what we can see in the middle here is that not only is there more, it's a different population. And this is important because Hawaii, one of our very own states, has the highest incidence of non-tuberculosis mycobacteria. which is, as it says, an infection of non-tuberculosis bacteria. And so this is work that we're just starting with Jen Honda in Colorado, and also some people at HVO, Tamar and Jeff, looking at the proliferation of bacteria in the Hawaiian environment, and then what knock-on effect that has for exposure and this high rate of disease that's observed. So by way of a quick summary, when it comes to ash, right, building this into what's the ash hazard, we're worried about ash because it has the potential to impact a lot of people. And ash in the environment stays around for a very long time. And people can be exposed through multiple different means. It can be environmental exposure, right, direct after the eruption. It can be through domestic exposure, cleaning up after the fact, for example, or occupational exposure. Occupational mining of volcanic deposits is a very big industry. And we know that ash can cause short-term problems, but we don't know whether or not ash can cause long-term disease, and or if ash can augment other diseases. And so I hope that I've shown some light on those last two factors throughout this talk, and why maybe we still don't know, but also why they're complicated and what we're doing about it. And those things being considering the mineralogy, considering the surface, and also considering processing. That is, what's in it, what's on it, and how that changes. And so even though we can delve in deep into one topic like this crystalline silica issue, that can be changed, and it's not a universal answer. So that's kind of what I'm doing here as a medical geologist. A medical geologist deals with the relationship between geology and health. And in order to do that, you really need to know both sides of this interface. And it's my thought that a medical geologist can, well, anybody really can characterize a hazard, right? Anybody can really run grain size and figure out what the particle size distribution of ash is. But I think it takes a geologist to be able to constrain the source of that hazard and where that hazard's coming from. And also that hazards are dynamic and that they can change. And without the fundamental research base that happens within the USGS, understanding those changes is difficult from the outside. And so I just wanted to highlight a few other cases where the USGS has been involved in this sort of response, much like this response to ash. And it's been going on for a very long time. And all of the bullet points up until the end are all response issues. Whereas this last bullet point, the health implications of disaster scenarios, this is a new thing for the USGS under this remit of the USGS multi-hazards demonstration project, where they're simulating earthquakes, storms, tsunamis on the coast of California, and then figuring out the repercussions of it. And that's all I wanted to say. So a quick thanks to all the people that I've worked with through the years. And also I'm in building 15 in room 1002 if you want to stop by to talk. Thanks very much.
So it does seem like there's still not a fire drill. They did delay it for us, so we do probably have time for a few quick questions, but I cannot guarantee the noise level in this room in 10 minutes.
Yeah, I'm just wondering, you talked about effects of diatomaceous earth and the health hazards of that. Is there a fundamental difference in the properties and or reactivity of biogenic silica or opal CT versus volcanogenic silica?
Yeah, it's a fine question. Yes, what those differences are difficult to constrain. And so if you take the case of diatomaceous earth, the industry isn't just one exposure. You have exposure from mining to processing to shipping and packing. And the exposure that you have through each of those stages is different, right? When you're extracting, you have it to unprocessed diatomaceous earth, which is, as you say, biogenic, non-crystalline. Through processing, two different mechanisms, with a flux or without, you get differences in the crystalline silica that's formed. And then, you carry that out, you have exposure throughout this whole process. And so when epidemiology studies say we see a high prevalence of disease in diatomaceous earth workers, there's no way to deconvolve that exposure, right? There's no way to say this person got disease because they were exposed to pre-processed diatomaceous earth. And so the way we go about it is to, like I showed you, mechanistically try to understand what those exposures look like, and therefore whether or not there's a difference between biogenic opal CT, semi-crystalline, so on and so forth. The common thought is that crystalline silica crystallinity is required for reactivity, but we don't see that in the experiments that we do. And there was also a paper out last year by some colleagues saying that amorphous silica can be toxic. So that's a big, round way to say I have no idea. But we're trying to consider every step of it.
So going back to the macrophages' response to the cristobalite, in the aluminum substituted case, was that a decreased response after correcting for the fact that it's been diluted with aluminum opposed to standard silica cristobalite? Or was that an inhibitory response? And if it was inhibitory, what's happening to the rest of the cristobalite? Is it just not being collected by the macrophages and just sitting on the lung cells?
So it's inhibitory. And we know that because we can take a particle, that we can take quartz, and substitute aluminum at the surface, and we abolish the response. So we know it's an inhibitory effect, and I've got some thoughts on why that's the case. But underlying all of this is that we don't know why crystalline silica is reactive. We don't know what about the mineral itself causes the reactivity. So then interpreting alterations like substitutions onto that baseline reactivity is quite difficult. To the second half of your question, it behaves as it otherwise would. It's internalized, and I didn't show it, but we have some data showing exactly how it's internalized, by the cell reaching out, grabbing it, and pulling it in. And that entire process works as it should and as expected. When it's inside the cell, this inflammasome stuff that I showed you it happens in a very specific part in the cell, and we see translocation of the particle into that region. So again, behaving as it would, it just doesn't cause the response. It just is inert once it's internalized.
Oh.
What's the status of research on volcanic aerosols and human health?
That it's up and running. I was in Hawaii, I guess 2 months ago now maybe, speaking with Tamar and Jeff and a couple other people, and we're getting that program off the ground. Not a whole lot is known. We know, much like we do with crystalline silica or other particles, we know a little bit about gas levels, concentrations that should be avoided. But in terms of volcanic emissions on humans, livestock, or plants, we don't really know yet the dynamic there. But that's what I'm here for, Maggie, and we're looking forward to it.
Any final questions? If not, let's thank David. And yeah, join us at the Tectonic Grill after, and let's leave the building quickly before we all get deafened.